Welcome to the Mind Blowers podcast. Today, we're diving into one of the most mind-blowing topics of all time, curing aging and the pursuit of immortality. Our guest is none other than Aubrey De Grey, the visionary scientist behind the concept of longevity escape velocity. The idea that science will soon be able to repair aging faster than we accumulate damage, effectively making aging optional. Our host, as always, is futurist Daniel Kafer. How close are we actually to longevity escape velocity? What scientific breakthroughs are needed to get there? In this episode, we'll break down the 7 key pillars that must be solved before we can stop and even reverse aging. Get ready for a deep dive into the future of human lifespan. Because the greatest revolution in medicine might not be fighting disease, but ending aging itself. Are we on the verge of curing aging? Let's find out.
Longevity escape velocity is, of course, not the easiest concept to understand. And in a moment, we'll hear Aubrey De Grey explain the 7 parts as best as possible. But first, I wanted to give you a quick overview of the 7 pillars of longevity escape velocity. I'll also try to guide you visually so you really get a grasp of how this works. We'll look at where we are today and make some predictions about how far we've actually advanced in each pillar. So what is longevity escape velocity? It's the idea that science and medicine will soon be able to repair aging damage faster than it accumulates. If we can keep up this pace, we won't just slow down aging, we could eventually reverse it. To do this, we need to tackle 7 key types of damage that cause aging, which have been identified by longevity researcher Aubrey De Grey. Each of these needs its own solution, and if we can address them all, we could radically extend healthy human lifespan. First, there's cell loss and tissue atrophy. As we age, we lose cells in vital organs like the brain and heart, which don't naturally regenerate. Scientists are working on stem cell therapy and tissue engineering to replace lost cells and keep our bodies functioning at peak performance. Next is extracellular junk, like amyloids and plaques, which build up outside our cells and contribute to diseases like Alzheimer's. Researchers are developing immune therapies to clear out this waste and restore normal function. Then there's intracellular junk, which is waste that builds up inside our cells, making them less efficient. Scientists are exploring ways to engineer special enzymes that can break down this waste and keep cells young. Another major issue is DNA mutations, which can lead to cancer. By improving gene therapy and developing ways to boost the immune system's ability to target mutated cells, we could dramatically reduce cancer risk. Mitochondrial mutations are also a problem. Mitochondria are the power plants of our cells, and when they break down, our cells lose energy. One promising approach is to copy mitochondrial DNA into the nucleus, protecting it from damage. Then there's the issue of cell senescence. Some cells stop dividing and refuse to die, releasing harmful signals that cause inflammation and damage surrounding tissues. Scientists are working on senolytics, drugs that remove these zombie cells and restore a more youthful balance. And finally, we have crosslinks in the extracellular matrix. Over time, proteins in our body become tangled, making tissues like arteries stiff and less flexible. Researchers are searching for crosslink breaking molecules to restore tissue elasticity and keep our bodies feeling young. Each of these areas is being actively researched, and some solutions are already being tested in animals and even in humans. If we can keep making progress, we could reach longevity escape velocity, where life extension is improving faster than we age, meaning we might never have to face aging the way previous generations did. So where are we now? How close are we to solving each of these 7 challenges? That's what I'm going to explore next. I'll show you how far we've come, where we still have gaps, and when we might expect real breakthroughs. But first, let's hear from Aubrey De Grey himself as he explains these 7 pillars in his own words.
Sure, yeah. So So the one I've just talked about is cell loss, where cells die and they're not automatically replaced by the division of other cells. And so stem cell therapy is the obvious way to fix that. We now, of course, also have this thing called partial reprogramming, which is essentially using the Yamanaka factors in vivo to make cells that are fairly stem-like already a bit more stem-like. And that's something that probably can stand in for stem cell therapy in a lot of places in the body, but not everywhere. I don't think you're going to be replacing your dopaminergic neurons using partial reprogramming. So we may end up being able to make the problem simpler using partial reprogramming, but we won't solve it entirely.
So let's make a quick recap. When it comes to cell loss and tissue atrophy, AI estimates that we've made about 60% progress in solving this issue over the last 20 years. And based on current advancements, we could realistically solve it within the next 15 years, making it one of the quicker aging problems to fix. Why? Because we already have promising stem cell therapies, tissue engineering, and regenerative medicine in clinical trials. Unlike more complex issues like mitochondrial dysfunction, where we need to rewrite biology at a deep level, cell replacement is more straightforward, just like swapping out old parts in a machine. And AI is playing a huge role here. It's accelerating stem cell research, helping us grow tissues in the lab faster, and even guiding personalized treatments for regenerating lost cells. With AI driving discoveries at an unprecedented rate, this could be one of the first aging challenges we truly conquer.
What's the next one? Next one, well, let's take cancer. I'd actually like to use a slightly more general term. I call it division-obsessed cells. So cells that divide when they're not supposed to. So thing about cancer, people often say, cancer is not just one disease. Different cancers are so different from each other. We need to tackle them in a very customized way. And certainly customized cancer treatments show great promise. We have this thing called CAR-T, which was developed not long ago now, only about a decade ago, and it's transformed a lot of things. The idea of using the immune system against cancer, or making the immune system work better against cancer than it normally does, has been around for decades and decades and made basically zero progress for God knows how long. And then suddenly there was this huge breakthrough, and almost simultaneously another breakthrough called checkpoint inhibitors, which essentially allowed the immune system to work better. And there's still a long way to go there, but it's going well. However, what we'd really like is an off-the-shelf, truly universal cancer therapy. And so we have to say, well, how can you have a universal cancer therapy when all cancers are different? Answer is that saying all cancers are different is an oversimplification. Cancers have distinctive features, but there's one thing that all human cancers have in common, and that is they all have a system for extending the telomeres, the ends of the chromosomes. If they don't, and the reason they have that is because a cell that doesn't have a system for extending the telomeres has its telomeres shortened every time it divides. And of course, the definition of cancer is cell division, uncontrolled cell division. So the cancer just will never get big enough, anywhere near big enough to kill you, without having to turn on some kind of telomere extension mechanism. And 90% of cancers in humans do it with this one enzyme called telomerase. So I've been interested in somehow disrupting the ability of a cancer to do that. And right now, we have a drug that does exactly that. It's an extraordinary discovery made by a great friend of mine, Jerry Shea, in Dallas, Texas, some time ago. Now in clinical trials in Australia, overseen by a company named Maya Biotechnology. It's a drug that basically, to use the technical term, it turns telomerase into a suicide gene. So to explain that in non-technical terms, basically what happens is that this drug kills cells that are making a lot of telomerase. And, it kills them quickly. So it's, I could go through the mechanism of how it does it, but the point is it does it really well. And especially in combination with a cancer immunotherapy, it's mind-blowing. So a big tragedy right now is that there's just this one clinical trial going on, relatively small number of patients for one type of cancer. We should be doing 100 clinical trials in parallel on different types of cancer, just using this one drug. And, so anyone who does want to throw, $1 billion at this problem rather than only the 20 million that I need, that's where I would put it.
Let's just recap. Cancer and unwanted cell division are among the areas seeing huge progress, and AI is playing a major role in solving these challenges. No one knows the exact timeline, but based on current breakthroughs in gene therapy, immunotherapy, and AI-driven drug discovery, it looks like we could eliminate most cancers within the next 12 years. Of course, this is just an estimate, but this is one of the fields where progress is moving fast, and every year brings new, groundbreaking treatments closer to reality.
So that's number two. And the reason I say division-obsessed cells is because there are things that involve the clonal expansion of cells in a subtler way, and that may be important as well. So there's something called myeloid skew. Let me explain briefly what that is. So in the bone marrow, of course, we have these stem cells for the blood that constantly generate the blood. And they are, there's lots of different types of cells in the blood. I mean, we say, obviously, there's red cells and white cells, and then there's platelets. But there's a lot of different types of white blood cell. And biologists group these cells, these many different types of cell that the blood has, into two major categories called lymphoid and myeloid. And it turns out, for whatever reason, that progressively through life, myeloid cells become more abundant, lymphoid cells become less abundant, and this is bad for you. You need lymphoid cells. People in Stanford, in Palo Alto, last year or year before, discovered a really cool way to reverse that process, essentially by killing off some of the myeloid cells and giving the lymphoid cells more opportunity to proliferate. And it seems to work really well, in mice anyway. So, yeah, this is another example. So I've only got through 2 of the 7 so far. The third one is another one that many people in the audience will be familiar with, which is senescent cells. So here we are talking about cells which, again, are bad for you, but they are not bad for you because they divide excessively. They're just bad for you because they hang around and they're toxic. They secrete nasty things. Now, these cells, people figured out, very much to my surprise, maybe 15 years ago, that there are drugs that can selectively kill these cells. And the first generation of those drugs were not selective enough. They were not really the real deal, but we're getting extremely good now at selectively killing off these senescent cells. They are things that, again, could potentially be eliminated by the immune system. The immune system does eliminate most of them when they arise, but not all of them. So again, people are also looking at ways to make the immune system better at doing that. But honestly, it looks like it probably won't be needed because drugs can do it instead.
When it comes to zombie cell treatments, this is probably one of the most advanced areas in longevity research. In the last decade, we've gone from early discoveries in mice to real clinical trials in humans, and the results are looking incredibly promising. Right now, scientists are testing senolytics, drugs that remove these harmful senescent cells, and early data shows they could improve bone health, reduce inflammation, and even slow aging itself. AI is also playing a big role here. It's helping analyze massive data sets to find new senolytic compounds faster, speeding up drug discovery and clinical testing. This means we're not just waiting for slow traditional research, we're actively accelerating the process. And the best part, we're already very close to having real effective treatments. Some senolytics are in late-stage human trials, and if the results hold up, we could see these therapies becoming widely available within the next decade. This isn't just theory anymore. This is happening.
So then, if we go inside the cell, we're talking, so far I've just talked about whole cells, right? So if we go inside the cell, then there are two major types of damage. The first one inside the cell is mutations in the organelle called the mitochondria. So all cells, almost all cells, have a lot of these things called mitochondria, which are responsible for extracting energy from nutrients and using it to do all the things that the cell does that need energy, like, building proteins and replicating DNA and so on. Mitochondria have their own DNA, and that DNA mutates much, much more rapidly than the DNA in regular chromosomes. We have to do something about that. And there are various approaches that might work. One way is to make copies of that DNA and stick those copies in the nucleus, in our regular chromosomes. And I've been trying to do that for about 20 years. It's our longest standing research project. And we've made a lot of progress, but it's very, very difficult. There are a lot of reasons why it's hard. And so we may never get to work, to be honest. Therefore, we'd like alternative approaches. And, essentially, most of those alternative approaches revolve around tweaking the way that the body recycles mitochondria. Even in cells that are not dividing, mitochondria are constantly recycled. Now, some of them are destroyed and other ones are replicated. And essentially, the balance between those two processes, the replication and the destruction, is what causes the problem. Mutants arise that somehow have a selective advantage and they expand, a bit like a cancer within a cell, if you like. And they take over the cell. So there are various ideas now for how to reverse that selection, so that the mutant mitochondria have a selective disadvantage and they never accumulate. Or even if they've already accumulated, that the expansion goes into reverse. But those are ideas that are at a pretty early stage of development right now.
Mitochondrial mutations and dysfunction are one of the more difficult aging challenges, and compared to other areas, progress has been slower over the last 20 years. The good news is that we now have more theories than ever on how to fix these mutations. Things like mitochondrial gene editing, allotopic expression, and mitophagy enhancement. But right now, most of these solutions are still in early stages, and we don't have a breakthrough yet. AI does play a role here, but its impact is likely to be medium rather than high, at least for now. Why? Because unlike drug discovery or diagnostics, where AI can rapidly process huge amounts of data, mitochondria involve complex biological processes that require physical interventions. AI can help model mitochondrial interactions, identify new potential treatments, and speed up research, but it can't directly solve the problem like it can in some other longevity fields. However, just because AI today can't fully solve this, that doesn't mean AI 5 years from now won't be able to. Future AI models will likely be far more advanced, capable of designing new molecules, running ultra-detailed biological simulations, and even optimizing gene editing strategies in ways we can't yet imagine. So while today's AI has only a moderate impact, more powerful AI systems in the near future could make a major difference in actually solving this issue. And here's the key point. We don't actually need a 100% perfect solution to reach longevity escape velocity. We just need a treatment that works well enough to keep us going until better therapies emerge. So even if we don't fully fix mitochondrial dysfunction in the next 10 or 20 years, we might have treatments that slow the damage, allowing us to stay ahead of aging until a more complete solution arrives. So while mitochondrial dysfunction is a tough problem, we shouldn't see it as a blocker to longevity escape velocity, just one more challenge that we'll keep improving on over time. And with the next generation of AI, we may find solutions much faster than we expect.
Okay, so the next one inside the cell is a nice simple thing to describe. It's just garbage, waste products. So the body makes a huge number of waste products all the time as byproducts of the things it needs to do. And nearly all of those things are destroyed as soon as they are created. The cell has a nicely diverse system for doing that. But there are a few things that the body doesn't know how to destroy, that simply accumulate really slowly. So evolution hasn't taken the trouble to invent ways to get rid of them, either by excreting them out of the body or by just breaking them down. And these things accumulate typically in a part of the cell called the lysosome, which is a kind of incinerator that can incinerate most things, but not everything. So basically, we need to improve the incinerator. So we've been trying to do that. It turns out that you can use the living world quite well in this regard. You can identify enzymes in bacteria or in fungi in the soil that already destroy things that we can't destroy. And then you can put the genes for those enzymes into cells and make them destroy things that they would not normally be able to destroy. This has ended up working really quite well for a material called A2E, which turns out to be the thing that causes macular degeneration, the number one cause of blindness in the elderly. This is a chemical that's created as a very occasional byproduct of the chemistry of vision, the chemistry of seeing stuff. And it accumulates in cells called the retinal pigmented epithelial cells at the back of the retina. So we've found enzymes that fix that. I passed this project on to a spin-out company out of one of my non-profits about 10 years ago, and they've continued to progress with it, and it's now working well enough that it's in the process of being sold to big pharma, so that's pretty good news. The other alternative, as I just alluded to, is rather than destroying the material in question, you can get the body to excrete it. And that's pretty difficult in the eye, but it's not so difficult in the bloodstream, and in particular in the circulatory system. So in the major arteries, we have material that's, again, not destroyed when we'd like it to be. And it is basically oxidized cholesterol. And so another group that used to work for me and now have a spin-out company is pursuing doing this, basically extracting oxidized cholesterol from the artery wall and causing it to be excreted out of the liver. So this is a big part of the gaps, if you like, in inbuilt damage repair machinery, the things that we need to fix.
Intracellular junk or lysosomal waste buildup is one of the harder challenges in longevity research. Over the last 20 years, we've made only about 40% progress, and the reason is simple. We still don't have a way to fully clear out all waste inside cells. Some of this waste, like lipofuscin, just accumulates over time, clogging up lysosomes and reducing cell function. Unlike senescent cells, which we can remove, these waste products aren't cells, so we need an entirely different approach. A major breakthrough would be finding enzymes or nanotechnology that can break down this waste and remove it safely. Right now, there are promising ideas like using bacterial enzymes that evolve to digest these materials, but we're not quite there yet. As for the timeline, at the current pace, solving this could take another 20 years. But AI might speed things up. AI is already helping to design new enzymes, optimize drug formulations, and simulate cellular processes much faster than traditional research methods. So while today's AI impact is moderate, in the next 5 to 10 years, more advanced AI systems could dramatically accelerate the search for effective lysosomal waste treatments. So while we are still far from a complete solution, we're heading in the right direction, and with AI-driven breakthroughs, we might not have to wait the full 20 years to see real progress.
So then finally, there are two types of damage that occur outside the cell. One of them is, again, waste products. So the most famous one of these is senile plaques in Alzheimer's disease. But the amyloid, as it's called, that accumulates there, accumulates in other parts of the body as well. It was discovered not all that long ago, 15 years ago maybe, that in the heart you get an accumulation of a different type of amyloid composed of a different protein called transferritin, which seems to be really bad for you when you get to be over 100. So we've got now ways to break that down. There's a special type of antibody called a catabody, which doesn't just bind to some antigen; it actually chops it up. It cleaves peptide bonds. So this is now very much in progress. Another spin-out company is working on that.
In the past 20 years, we've made real progress in tackling extracellular junk, especially amyloids and plaques linked to Alzheimer's. One major breakthrough was beta-amyloid immunotherapy like aducanumab, designed to clear plaques from the brain. Another big leap was the Alzheimer's in a dish model, which lets researchers study plaques in lab-grown human neurons, speeding up drug testing. But challenges remain. These treatments are expensive. Their effectiveness varies, and they don't fully cure the disease, just slow it down. Reaching 100% success is tough, and we're still unsure if we'll ever completely eliminate amyloid buildup. AI, however, is starting to change the game. Right now, it's helping scientists design better drugs and analyze clinical trials faster. In the future, AI could make treatments more personalized, improve early detection, and even help create entirely new therapies. While AI won't solve this overnight, it's making progress happen faster than ever. So while there's still work to do, we've come a long way, and with AI in the mix, we might see even bigger breakthroughs sooner than expected.
And then finally, there is cross-linking. So again, coming back to the arteries, the cholesterol I was mentioning is the cause of atherosclerosis, which causes heart attacks and strokes and so on. But we also have this thing called arteriosclerosis, the hardening of the arteries, which leads to high blood pressure and lots of knock-on effects of that. And that's basically a loss of elasticity of the arteries. And it's the same chemistry that goes on there that causes wrinkles in the skin, loss of elasticity there, and also, for that matter, presbyopia, so long-sightedness, inability to focus on things close up. It's all to do with chemical reactions between proteins and sugar in the circulation. And these chemical reactions cause new chemical bonds to form between proteins that are supposed not to be bonded together and supposed to be able to slide against each other. So this is where the stiffening comes from, the loss of elasticity. And so now the goal is to develop ways to break those bonds. And it's going quite well. Again, there's a spin-out company working on it that comes from the work that we funded a long time ago. So yeah, all of these things are moving forward, but it's still, in many cases, a long way to go.
When it comes to crosslinks in the extracellular matrix, we've made some progress, but it's still one of the harder challenges in longevity science. Over the last 20 years, we've identified glucose pain as a key aging-related crosslink, developed self-healing hydrogels, and uncovered how tissue stiffness affects diseases like cancer. But right now, we still don't have a way to fully reverse crosslinking in humans. A full solution could take 20 to 30 more years, since safely breaking down crosslinks without damaging healthy tissue is tricky. AI is already helping by simulating how crosslinks form and designing potential enzymes or molecules to remove them. In the short term, AI will speed up drug discovery, and in the long run, it could help us develop targeted crosslink breaking therapies. But here's the key point. We may not even need a 100% fix to reach longevity escape velocity. If we can just reduce crosslinking enough to restore flexibility and function, we buy time for even better solutions down the road. That's where AI comes in. Right now, its impact is moderate. It can speed up research but can't physically break crosslinks. However, future AGI-level AI could completely change the game. A truly intelligent AI capable of fully understanding biochemistry and cell dynamics could design entirely new enzymes, molecules, or even nanotech solutions beyond human capability. What might take decades could happen in just a few years. So while we're not there yet, progress is happening. And with AI's help, we may get there much faster than expected.
The research you do in the mice,
do you focus on one area at a time in these 7 or all of them at the same time?
The answer to that question has changed in the past couple of years. In my previous organization, Sense Research Foundation, and the one before that, the Mitochondrial Foundation, I was doing one at a time. And in particular, I was focusing on the most difficult ones, the ones that there had been least progress because they needed to catch up, right? And also because nobody was working on them because they were the harder ones. And, in academia, you tend to go after the low-hanging fruit because you need to get lots of publications in high-profile journals in order to, get promoted and get your next grant application funded and so on. So it fell to me as the leader of a non-profit that was funded entirely by philanthropy to do the things that other people were not in a position to do. So that was all great. And as I say, there's been plenty of progress, spin-out companies and so on. But now at LEV Foundation, we are moving on to stage 2, the putting them all together. Because, of course, any divide and conquer approach, which this obviously is, that has those 2 phases. You develop the components and then you put them together. And in most engineering contexts, that second part, putting them together, is the easy part because you pretty much know that when you put things together, they're going to work once the individual components do. But in a living organism, you don't know that at all because you don't really understand how the thing works at all. So you may put them together and get surprises. You may find that things that individually are beneficial cancel each other out, things like that. So we're at that stage now. We're actually not taking the hardest things, the things I used to work on, because those things are really not ready. They're not really working well enough, even in mice, to be combined with other things. No, we're working on the low-hanging fruit, the easy things, because those are things, even though I wasn't working on them before, other people were, of course, working on them and making progress. And so now there are at least well over half a dozen ways to repair this or that type of damage in mice, and you actually get a little bit of life extension. Simply because of these knock-on effects I was mentioning. You have, improvements in basically everything if you improve one thing a lot. And so, of course, it makes sense to try and put these together and see whether we can get additive effects. And that's the experiment that we've been doing for the past 2 years. Now, it's the first time anyone's done an experiment like this. And it was a very big experiment. Started out with 1,000 mice because we wanted to do lots of different combinations. And, you've got to have like 50 mice of each sex in each treatment group. So 20 treatment groups. Basically we got the result we wanted, that we are seeing additive effects from these interventions. So, we need to keep going. The next experiment we want to do will be even more expensive because we want to put like 8 interventions together rather than only 4, which is what we did this time. So we found ways to save money as well. So it's not going to be twice as expensive as the previous experiment, but it'll be 1.5 times expensive. The previous one cost about $3.5 million. The next one's going to be 5 or 6. That's what I'm aiming to try to do.
So when will we actually reach longevity escape velocity? In my opinion, there are 2 major factors that will determine this. The first is simply how much investment we can pour into longevity research and the areas that Aubrey De Grey has highlighted. The more funding and focus we put on solving aging, the faster breakthroughs will come. The second factor is artificial intelligence. Right now, AI is mainly helping to speed up research, optimizing drug discovery, and assisting in clinical trials. But the real question is, when we reach more advanced levels of AI, can it do more than just assist? Could it actually develop new solutions or even create biological simulations so advanced that we no longer need years of animal testing to validate treatments? A few people are starting to put timeframes on when we might get there. Ray Kurzweil is the most optimistic, believing we could hit longevity escape velocity in just 5 years. Then we have Aubrey De Grey, who estimates 12 to 15 years, and David Sinclair suggests it might take around 20 years. Of course, these are all projections, and the real timeline will depend on how quickly we make breakthroughs in both biotechnology and AI. At this moment, we're not quite there yet, but who knows what AI may bring to the table in the coming years? If these two factors, funding and AI advancement, align at the right pace, we could be looking at longevity escape velocity far sooner than most people expect. If you are interested in more videos on longevity, check out the playlist link in the description.